So good afternoon and welcome to Jamal from Elicera. Elicera released a very interesting message the 13th of September regarding two conferences they're going to participate in during the autumn. Welcome, Jamal. Will you please elaborate on what is going on?
Thank you, Klaus. Yes, and thank you for this opportunity to present the iTANK platform. So, the news that we released in September is with regards to two conferences where we will present data on our iTANK platform, which is a platform that we use to arm our CAR T cells and oncolytic viruses that we have under development. So the first conference will be now in October, on October 19th, and the second presentation of data will be in November 2nd. We also have submitted a manuscript on the iTANK platform that we hope will be published sometime this year. So just briefly on what the iTANK platform is. So Elicera is a company that develops oncolytic viruses and CAR T cells. And we have quite a broad pipeline of drug candidates, so 4 in total. And all of them are very powerful and have a great potential to treat cancer. But I would actually say that the iTANK platform is perhaps the most promising and valuable asset that we have today. So this presentation will focus on that platform, the iTANK. But before going into that, I will just give a very brief introduction to the company. So Elicera Therapeutics is a company that works in the field of cell and gene therapy, and we're focusing on immune oncology, meaning that we develop therapies that uses the patient's own immune system to fight cancer. And in this case, it means that we develop CAR T cells and oncolytic viruses. The company is listed on Nasdaq First North Growth Market here in Sweden, and you can find the share under the ticker ELIC. 4 drug candidates under development. The most advanced is in a phase 1/2 study in neuroendocrine tumors. And our drug candidates have been genetically modified so that they elicit multiple modes of actions in one drug candidate. And that's where the strength lies in what we do. And this is based on the iTANK platform. We have a team that has relevant experience within both board and management. So we have clinical experience from both CAR T cells and oncolytic viruses. Our CSO and co-founder, Professor Magnus Hassan, was actually part of the very first CAR T cell study ever performed in Europe. And myself, I have experience from acting co-founder and CEO of another immune oncology company called Immunicum for almost 10 years. So short on our pipeline, you see here that we have 4 drug candidates under development, the most advanced in neuroendocrine tumors. ELC-201 is our so-called next generation oncolytic virus. We have armed also this one with the iTANK platform, 301 and 401 as well. ELC-201 is an oncolytic virus that generates a combination of 3 mode of actions in one product. ELC-301 is our first CAR T cell that will enter clinical studies planned next year. And it's in liquid tumors where the first indication is B-cell lymphoma. And we also develop ELC-401, which is in development for a range of solid tumors. Not all of them, but initially we will target glioblastoma multiforme. But we also see a potential in, for example, colon cancer, melanoma, head and neck cancer, and pancreatic cancer, among others. And then ELC-001, which is what we call then the iTANK platform that we use to arm 201, 301, and 401. And important to mention is also that we think that this iTANK platform can be used to also arm the product candidates of other CAR T cell developers. So it's a technology platform that we believe we could capitalize on in the short term by licensing deals. And it's something that we intend to investigate during the fall here. I also want to mention before presenting the iTANK platform that we haven't really marketed the iTANK platform to the industry. So the first conference here where we will present some data is the first time actually that we publicly will talk about the iTANK platform and data from that. And with the publication later this year, this will give us a good foundation to find potential partners that could be interested in this platform.
So just very briefly, Jamal, this
Sure.
is really great potential also for running cash flow while you do your own project management. And it doesn't stop. It doesn't stop here, as you mentioned. So one thing is you can license and partner with this, but you can still discover new ideas yourself.
Correct. So I will talk a little bit more about this potential later in this presentation. But exactly as you say, this platform we think could be partnered on a non-exclusive basis, meaning that we could generate several partnerships for this specific technology.
Good.
So before going into that, I just want to give you a short background on what a CAR T-cell is, to give you some understanding. CAR T-cell therapy is one way to treat the patient by the immune system. So immuno-oncology encompasses many different types of therapies, and CAR T-cell therapies is one of them. It's also perhaps one of them that's considered the most promising one. So ASCO, which is one of the world's largest cancer organizations, named CAR T-cell therapy Advance of the year 2018. And they did this because it has been shown then that CAR T-cell therapy could actually cure many patients that were previously untreatable with different types of liquid tumors. So they had no treatment alternatives. They received CAR T-cell therapies, and all of the tumors were eradicated. So it showed proof of concept for this type of therapy. So what it is basically you take out blood from the patient and you sample out the T cells from the patient. And the reason you do this is because T cells are the immune system soldiers. So you take out the T cell and then you genetically modify them so that they express a chimeric antigen receptor, a CAR molecule. And this CAR molecule then recognizes certain targets on the tumor cell, antigens, which it binds into and starts killing the tumor. So you multiply this in test tube, you re-inject them into the patient, and then they will find all these tumor cells that carry these antigens that the CAR molecule is directed against. As I said, this works very well in liquid tumors, but there are still some challenges in solid tumors. And the two main reasons for this is that in a solid tumor situation, you have a very hostile tumor microenvironment, which inhibits the effect of the CAR T cell. And this hostile tumor microenvironment you don't have in the same way in a liquid tumor situation where you have blood cancer. So it's more easy for the CAR T cell to have an effect in different types of blood cancers. Also, there are very good targets in different blood cancers, mainly CD19 and CD20. But when you look at the solid tumor situation, it's difficult to find a good target for a CAR T cell that's not also expressed on healthy cells, because you want the CAR T cell only to target cancer cells and not healthy cells, of course. So there's an antigen heterogeneity that you have to take into account in a solid tumor situation. And we developed two CAR T cells that can address both of these problems. And the reason that they can address both of these problems is because they have been armed with the iTANK platform. And we can, through this iTANK platform, both inhibit the hostile tumor microenvironment by creating a very pro-inflammatory environment that actually triggers the immune response and that makes the CAR T cell stronger. But we can also generate a second mode of action through activation of the patient's own innate T cells against a whole repertoire of different targets on the tumor cell, not only the one target that the CAR T cell is against. So to further put this in perspective, you can say that CAR T cells have been developed in different generations. So first generation CAR T cells, they were shown to have an insufficient proliferation. They had a very brief T cell survival, and they also had an inadequate and insufficient immune activation. So because of this, then you created better CAR T cells in second generation, and then third generation, and then finally now fourth generation CAR T cells. And what we develop is, you can say, an optimized version of fourth generation CAR T cells. And I will just explain shortly why this is so. But the general difference between fourth generation CAR T cells and other generation CAR T cells is that fourth generation contains a transgene that codes for a substance that can trigger the immune response. And usually it's a cytokine transgene that codes for IL-12 that can trigger the immune system. And the way we differ is that we don't code for a cytokine transgene. We put in a gene that codes for a neutrophil activating protein, short for NAP. And when NAP is activated, it actually releases over 20 different cytokines and chemokines, not only one, that we believe give a much broader and more effective immune response against cancer. We can also mention that 70% of the CAR T cells under development are still second generation, and only 16% are fourth generation. And of these 16%, most of them we believe are being developed in academia. We've actually only been able to identify one other company that develops a fourth generation CAR T cell, and it's a Japanese company called Noile Immune Biotech that I will talk a little bit more about shortly. Before doing that, I just want to go into a slide that gives you quite many details on the mode of action of the iTANK platform. So this is to try to explain how it works and why we can use it to generate a second attack of the immune system by the patient's own CD8 positive killer T cells. So if you look here in number 1, furthest to the left here, you have a CAR T cell that has been armed with the iTANK platform. You see the NAP gene here. So when the CAR T cell binds into a tumor cell, NAP gene is activated. And this releases cytokines and chemokines that triggers an immune response. And the first thing that happens is that the patient's immature dendritic cells will come to the tumor site. And what these immature dendritic cells do is that they pick up tumor antigens, which are tumor fragments from the tumor cell that has been attacked by the CAR T cell. And the reason this is important is because dendritic cells are very potent antigen-presenting cells. So they will pick up these targets, not only one, but the whole set of different tumor targets that have been released by the killing of the CAR T cell and the tumor cell, which means that the activated dendritic cells will then go into the lymph nodes and present these antigen targets to the CD8 positive T cells. And then they become activated against these targets. So you will have CD8 positive killer T cells that are activated against a whole repertoire of different tumor targets, tumor antigens, not only the one target that the CAR T cell will find. So you will get you can say a combination of 2 attacks on the tumor cells, one by the CAR T cell and the second by the CD8 positive T cells on the whole set of different antigens. And this is what we're trying to clarify with this slide here. So normally when you treat patients with CAR T cells, you only have the CAR T cells that target the tumor cell. And if you look closely on the tumor cell here on the picture to the left, you see that we have some of the fragments of the tumor cell where we have different colors. And this is to show you that the tumor cell contains different antigens, not just one. And in picture 2 here on the right, you see that we have CD8 positive T cells that bind into different targets with different colors here to exemplify then that you have a much broader attack on cancer. So this is the big advantage of using the iTANK platform on CAR T cells. And this is what we do with our own CAR T cells. And this is what we believe we can do for other CAR T developers.
So companies in second generation is actually able to apply your technology or the iTANK platform.
Yeah, correct.
And then they can advance into the fourth generation NAP technology.
Correct. So that's exactly correct. So what the reason that we say that our CAR T cells that we have in our pipeline are fourth generation is because we use this iTANK platform to arm them with this NAP transgene. So whenever we would use this same technology to arm other CAR T cells with this NAP transgene, that would in essentially make them also fourth generation CAR T cells. So they could use their same technology that they are working on, their own CAR T cells, but then also just add this transgene with NAP to make them even stronger, more potent, and to elicit this second mode of action. So that's correct, yeah.
Yeah.
So we'll present some preclinical data now in October, in November, and also, of course, more extensively in the publication that we expect to be released later this year. I will just show you that we have treated the mice where we have injected these mice with CD19 positive tumor cells, 50%, but also 50% of these tumor cells that don't have the CD19 antigen expressed. And then we've treated them with conventional CD19 CAR T cells, and then CD19 CAR T cells that have been armed with the iTANK platform, or the NAP here. Mm-hmm. And if you remember then, if you have a CAR T cell that targets CD19, they will only, of course, bind into the CD19 antigen, meaning that if you have a tumor cell that lacks the CD19 antigen, the CAR T cell won't be able to have any effect. Nope. So if you look here at the figure on the right here, you have tumor size and the survival of these mice. And the CAR-NAP T cells are the red graph here, and the blue is the conventional CAR T cells, and the black is the placebo, you can say. Mm-hmm. So as you see, with a conventional CAR T cell, it basically doesn't have any effect at all. And this is because they can't bind into the tumor cells and start killing them, since these tumor cells, 50% of them don't have the CD19 antigen. But if you treat them with our CD19 NAP-enforced CAR T cells, then you see that there's a shrinkage of the tumors. And this can then be deducted to the second mode of action here, then the activation of CD8 positive T cells against other targets than CD19. And we see the same thing when we study survival of these mice. So in the figure here, farther to the right, you see that the conventional CAR T cells don't seem to be able to really prolong the survival of these mice. But when you treat them with NAP-enforced CAR T cells, you see that it prolongs the survival of these mice.
Yeah, that's more than double. That's more than double, yeah.
Yeah, so it's quite a promising proof of concept in preclinical studies for this concept here. So this slide is only to show you that there are different ways of working with a CAR T cell. So you can either work with the T cell in itself, or you can work with the CAR molecule. You can work with boosting or arming the CAR T cell, which is what we do, and the other company that we've identified here, Noile Immune Biotech, what they do in their Prime T platform. Or you can work with manufacturing. So I just wanted to show you that there are different ways of doing this, and that fourth generation, they belong to this boosting and arming of the T cells, and that the only competitor in this area that we've seen is this Japanese company and their Prime T platform. So it's an interesting example, I think, because the Prime T platform, the description of it is basically exactly how we would describe our iTANK platform. The difference is that the Prime T platform, they use 2 cytokines, while we use the NAP protein that releases over 20 different cytokines and chemokines. So we believe that this has a greater potential to give a more broad and better immune activation than the Prime T. What's also interesting is that if you study Noile Immune Biotech, you can see that the recent years here, they've been able to generate 8 different licensing and collaboration agreements with other CAR T cell developers around their Prime T platform. And this just shows you that the industry would be very interested in this type of technology, because everybody's struggling with the efficacy with CAR T cells in solid tumors. So in liquid tumors, we already know that it has a good effect. There's still more work to be done, and I don't have time to go into that in this presentation. But in the solid tumors, everybody still has struggles, and the iTANK platform could help out in this area. And the final slide I have here is not an exhaustive list of companies that are working with CAR T cells. We've taken some of the most prominent ones, both bigger and smaller companies. And we've shown you here in this slide what they are focusing on. And you see they either focus on 1, 2, or 4. But none of them are focusing on arming their CAR T cells with a transgene that activates this second mode of action. Number 3 here. So to go back to your question there, Klaus, yes, all of them could, we think, in theory at least, could have benefit of using the iTANK platform. And these are the types of potential partners that we think could be interested. So with this, I thank you for the opportunity to present the iTANK platform, and I look forward to coming back to you with more information once we've had the presentations and the publication out.
Thanks a lot, Jamal. Very interesting, and very interesting that you're able to actually grow the market, not only as a company, but as an industry. But that will also lead us to, hopefully, we can treat patients sooner than expected. So with that said, Jamal, thanks a lot, and we look forward to host you later on in autumn. Thank you.
Great, thank you.